Okay, folks, it's 7 after 8. Wow, instead of 5, so we'll start now. Even let's say there are 8 of us here, so it's like 8 out of 10. Close enough. All right, so today I'm starting mutation. What we'll be doing is we will looking at, first of all, we'll define what a mutation is, and then we'll look at the major types of mutation as well as the relevance of mutation to evolution as well. Okay, should already be seeing my screen. So let's begin. So what is a mutation? A mutation refers to any change at all in hereditary material, okay? Mutations are usually spontaneous changes to your hereditary material. So simply put, a mutation happens from one generation to the next. And by that I mean, it will not be present in one generation and it will simply appear in the next generation. Okay? So that's the first thing. Most mutations that occur are what we consider to be neutral. They're not good, they're not bad, they're just there. Okay? Though a small percentage of mutations can be harmful or they can be beneficial. Generally speaking, beneficial mutations will be amplified within a population and the harmful ones will be diminished. Once again, this is generally speaking, right? So in multicellular organisms like ourselves, you're pretty much going to find two categories of mutations, okay? One of which is going to be much more important than the other. We have what we call somatic mutations and germline mutations. You should already be aware of what the word somatic means, right? As in somatic cells. One second. Okay. Why did the background color change? Give me a second here, something funny does happen. Okay. All right, there we go. So, where were we? Yeah, types of mutation. So two major groups are somatic, as in somatic cells, right? Now, somatic cells refer to non-sex cells, right? So non-sex cells. Whereas germline mutations, no surprise, are going to refer to sex cells. Which of course means germline mutations end up in your sperm and of course the ova, okay? So egg cells, sperm cells, of course, are ova. They will carry germline mutations, all right? Now, this is very important with regards to evolution, okay? If you have a somatic mutation and that mutation sits only in your normal body cells, your somatic cells, let us say it is a positive mutation. Let us say the mutation allows you to, I don't know, fly, even though that's impossible for one mutation to do that. But let's say it does. It's the most awesome mutation in the world. If that mutation only lies within your somatic cells, you cannot pass that mutation on to your offspring. And therefore, your awesome ability to fly is an evolutionary dead end. It cannot leave past you. It's pointless. Whereas if it occurs in your sex cells, even if you do not have the mutation yourself, you can fly. It means you can pass on the ability to fly to your children, and they can pass on the ability to fly to their children, and all of a sudden, your species begins to evolve. Okay? I hope you see why this is important, right? So somatic cell mutation only affects your non-sex cells. So the mutation will only affect that individual that has it. But if the mutations become germline mutations, then they end up in your sex cells, which of course, once again, means you can pass those on to your offspring, right? Okay. So remember, somatic mutations arise in the somatic cells and are passed on through the process of mitosis. So normal non-sex cells, they replicate, of course, but it still means that it's only your cells, right? The effect of these mutations depend on the type of cell in which they occur and the developmental stage of the organism. If it occurs early in development, larger number of mutated cells will occur in the organism. Okay, so that should make sense to you, right? Because remember, we start out as one zygote, and that zygote splits into two, and into four, and into eight, and it will continue onwards, right, until you form a fetus. If you have a mutation in one somatic cell, and that mutation began after the second division, right? So your one zygote becomes two cells, and then one of the cells has the mutation. It means, of course, that half your body cells will contain that same mutation, right? All right. Now, germline mutations, as it says, they occur in the cells that produce gametes. And of course, you can pass these mutations on to future generations, right? Now, usually when we say a mutation, we refer to germline mutations. Why? Because once again, if they occur in your body and you can't pass them on, they're not particularly useful, right, to the species. That's why we normally, when we say mutation, we talk germline mutations because those are relevant. Right? Now, as it says, the term mutation was derived by this fellow here, De Vries, in 1901. But as usual, we're not going to waste too much time on the people, just on their work, right? Now, the types of mutation that were initially discovered were chromosomal mutations. Because, of course, chromosomes are relatively large, you know, for cellular component, you know. I mean, obviously, these are going to be subcellular components, so they're tiny, but still, chromosomes are reasonably big in comparison to a lot of other things there, right? So they could look at the chromosomes under the microscope and they could see physical differences in the way the chromosomes looked. And that way they could say, hey, this chromosome number 5 here should look this way, but this one here look funny. It's too short, it's too long, it has bands in the wrong place, you know. They could pick up the differences by just looking at them. And now some mutations were point mutations, which means, of course, they occur at very specific areas of the chromosome, and they don't affect the rest of the chromosome, right? Now, initially, they did not separate chromosomal mutations and point mutations, okay? So just call everything mutations. Of course, now we do separate them. So we're going to discuss chromosomal mutations and point mutations differently. So remember, chromosomal mutation affects the entire chromosome. A point mutation affects a tiny part of the chromosome. Now we normally refer to this part as the gene, okay? All right. Now, obviously, mutation is going to have to occur within the genetic information inside the cell. So no surprise, we need to talk about DNA, okay? As it says here, DNA is a highly stable molecule that replicates with amazing accuracy. I mean, it makes one mistake in 10,000, so that's pretty good, right? Some errors of replication do occur, of course, and a mutation is defined as an inherited change in genetic information. Remember, the inherited part is because we're talking about germline mutations normally when we say mutation, okay? All right, so as to the types of gene mutation, so there are several different ways of classifying gene mutations, right? You can classify them based on the molecular nature of the defect, right? So basically, you're looking at the basis, right? Or you can look at the mutation in terms of the effect changes in the basis have on the phenotypes. All right. So when you change a base here in the DNA molecule, it alters the structure of the protein that is produced by the gene. And then now we can see how the altered protein impacts the organism, okay? You can also look at mutation in terms of the causative agent. So is it radiation? Is it chemicals? You know, what is the mutagen? If you ever seen Teenage Mutant Ninja Turtle, you know what, you've heard the term mutagen before, right? Now, so when you're looking at mutation with regards to the molecular nature of the defect, here are the types of mutations that you can expect to see. We have base substitution. Now, you guys should already be aware of the major bases, right? That you find. May I assume that is correct? Can I assume everybody knows what's it, A, C, G, T, U? Can I assume everybody knows this? Daniel, what does A stand for?
Um, um, I don't remember.
Then why y'all sat there and more quiet when I asked if you didn't know what this is? Kalicia, what does A stand for? Kalicia Swebe, what does A stand for?
Sir, I don't remember.
Once again, why y'all sat there not knowing?
Sir, is it adenine?
Yes, it's adenine. I can't remind you of things if I don't know, you don't remember them, people. Adenine, cytosine, guanine, thymine, and the last one here, uracil. Right? Hopefully this looks familiar to you, right? You should have done this in college bio, right? So,
Yes, sir.
okay, good, lovely. So this is what when I say base substitutions, we're talking about these bases, okay? And we also have what we call insertion and deletion mutations as well, okay? So let's start by talking about what these things are. So remember the bases, A, C, G, T, U, right? So in a base substitution, very simple. One of the bases in a DNA strand is altered. So let's see here. We have, by the way, can anyone tell me if this is a strand of DNA or RNA? Who can tell me if this is a strand of DNA or RNA, please?
DNA.
All right, one person who's a DNA. Was Daniel?
Daniel.
You are correct, Daniel. Who can tell me how they know this is DNA?
There's no uracil, sir.
There is no uracil, Kelisa. Thank you very much. Wonderful. So we know this is not RNA. It has to be DNA, right? Now, when we look at the bases of DNA and RNA, we read them as a group of three. So we take a string of the bases and we group them in threes. And the reason we group them in threes is because it takes three bases to code for an amino acid, okay? So in DNA, we call the threes triplets, right? Triplets in DNA. In RNA, we call them codons, but we're dealing with DNA here, right? Okay. So we have here a section of DNA strand. We have The DNA strand, of course, is a double helix. We have two parts. We have the sense strand and the anti-sense strand, as I'm sure you all know, right? So the strand that is being affected, of course, is the first one. So this is the original DNA up here, right? And this is the DNA after base substitution. So if we look here at the triplet number 3, obviously it's in red, so you can see it. We go from GTA to GCA, okay? So it's called a substitution because you're substituting one of the bases for another one. Easy as that, okay? So you swap out a base, put in another one, that's your substitution, right? And it shows, of course, what happens after you go through DNA replication, right? Now, whether or not a mutation like this is significant is going to vary depending on where or whether or not this mutation here results in a change in the amino acid. Sometimes you can have a change in a triplet and that change does not actually affect the amino acid that is asked for, right? And that, of course, is a missense mutation, but we'll talk more about that later. Just remember right now, substitution mutation, you're swapping one of the bases for another base in the codon, in the triplet, sorry. Simple as that, right? Now, you should also remember the terms purine and the terms pyrimidines, right? You should know purines, and you can correct me if I'm wrong, are double ringed, something like that. I'm not going to put in the other things. And of course, pyrimidines are single ringed bases, right? So hopefully remember that from college bio, right? And purines bind to pyrimidines. That should also sound familiar to you. So your purines are, and hopefully you remember this, are adenine, one second. Strange. Anyway, are adenine, guanine, right? And your pyrimidines are going to be the rest of them. So cytosine, well, start with thymine, then uracil, and of course, cytosine. Now, what is important about purines and pyrimidines is that they bind to each other, okay? Purines like to bind to pyrimidines. Now, adenine in particular loves to bind to thymine, okay? This happens, as you know, in DNA, right? But adenine can also bind to uracil, but that only happens in RNA, okay? And guanine, of course, guanine is loyal, so guanine only binds to cytosine. So far, everything I've said should sound familiar to you, right? So purines bind to pyrimidines, guanine binds to cytosine. So guanine is a purine, cytosine is a pyrimidine. Adenine binds to thymine in DNA, and you're still in RNA, right? So basically, this is what we have going on here. Now, it mentions here that substitution is going to be of two types. So you can have a purine that is replaced with another purine, okay? So adenine can be replaced by guanine, or the other way around, sorry. Or you can have a pyrimidine, which is replaced by another pyrimidine. Okay? Real simple. So substitution, you swap out, when you swap in an A, you can put a T there. Right? Or is the entire thing is gonna be problematic. All right, we'll talk about, of course, what happens if that does happen, because it can potentially happen. So remember now, you have transitions, right, where you're swapping with the same group. So swap a purine with a purine, or you swap a pyrimidine with a pyrimidine. That's a transition. Now, when you do it the other way, as I was mentioning a moment ago, when you swap a purine with a pyrimidine, this normally causes all sorts of problems, which we'll talk about. So you swap a A with a C, or A with a T, G with C, G with T, right? Or the other direction, C to G, C to A, T to G, T to A, are transversions. So remember, if you're swapping within groups, it's a transition. If you're swapping between the two groups, it's a transversion, okay? In both cases, that's substitution. Now, obviously, there are more pyrimidine bases than purine bases, right? And now, since you're swapping in between pyrimidines and purines, it means you have more possible combinations, right? Because remember, if you're swapping pyrimidine to pyrimidine, you basically can do 3 swaps, right? So the number of possible transversions is going to be twice the number of transitions, simply because, once again, you have 2 groups, one with 3 bases, one with 2 bases, and they're interacting, versus having a group of 3 and a group of 2 separate, right? Okay. But transitions are usually more frequent in vivo. Okay. Does anybody know what vivo means? All right. Do you know what in vitro means?
No, sir.
Okay. In vitro equals in glass, and in vivo equals in life. So if you've heard of in vitro fertilization, for example, they take the egg, they take the sperm, they combine them in a petri dish, which is normally glass, and then they take the fertilized over and they insert them in the mothers. Right? Normally it's like 30, 40 of them they put in one time, hoping that one will catch. But the idea here is that you start the process in a glass container, therefore it is in vitro. In vivo means in life, which means it happens in the fetus growing inside its mother. Okay? So in vivo versus in vitro, right? So if you make any changes to a living organism that's inside its mother, it is in vivo. Any changes occurring inside, outside the mother is in vitro. Okay, so that's it for substitutions, the basics of it, right? Then now, let's talk about insertions and deletion. Now, obviously, the term insertion means you're adding something new, you're sticking something in, right? When you insert yourself into people's conversation, you're putting yourself somewhere you don't belong. And deletions, of course, you're removing something. So it should come as no surprise that you're adding and deleting bases, okay? Now, as it says here, it's the second major class of gene mutation, addition or removal, respectively, of one or more of the nucleotide pairs. So once again, the bases, right? Usually changes that the reading frame, altering all amino acids encoded by the codons. This is important because another name for insertion and deletions is frame shift mutations. And I'm going to explain why that is so much worse than substitution in a minute, okay? Just be aware, remember, insertions and deletions, you're adding bases or you're removing the bases, okay? And by adding and removing the bases, you affect all the bases that come after the one that you've changed, right? Now, that said, you can in addition to looking at the bases, we can also look at mutations in terms of the phenotype, as I mentioned before. And it mentions the most common phenotype in natural population of an organism is called the wild type. We've done wild type already, right? That's normal, right? So whatever phenotype is, whatever genes generate the wild type phenotype would be considered the norm. Anything outside of that would have to come from a mutation. For example, if I say to you, what was the original eye color of human beings? What answer would you give me? So eye color is a phenotype, right? Go ahead, Blessing.
Brown, sir?
Brown, you say?
Yeah, I said brown.
Okay.
Yeah.
Now, first of all, people don't really have black eyes, right? If you shine a light into the eyes of a person with very, very dark eyes, it still looks brown because melanin is brown. If you have a lot of melanin, it can appear to be black, but it's just extremely dark brown. So Blessing is completely correct. Brown eyes is the wild type. It is the first type. It's the natural type. So blue eyes and green eyes and gray eyes and very light brown eyes are all mutations of the wild type. Just like the original hair color was black. Well, technically, even the hair color, the black hair color, is not really black. This is extremely dark brown once again, because this is melanin that colors your hair as well. Which is why the same thing that can bleach your skin can bleach your hair. One of the reasons, anyway. Right? Now, remember, wild type equals the normal type that is you find most people in the population have. Everything else beyond that is a mutation. Now, a forward mutation is going to be one that alters the wild type. That means that the forward mutation spreads within the population so rapidly, well, so pervasively, I should say, and eventually the original wild type disappears. Okay? So, for example, when Africans left Africa 75,000 years ago and went up north, they still had dark skin, right? They would still have had far more genes for darker skin color than they would have had for lighter skin color. Remember when we did multiple alleles, right? You have multiple genes that determine your skin color. The more dominant ones you have, the darker your skin will be, right? You all should remember that. As they went further and further up north, the mutated gene for lighter skin color became more important because it led to, of course, better survival of the children. And over the course of thousands of years, the original darker dominant dark skin would have disappeared from the population, and then the mutated light skin gene would have become predominant in the population. So in Caucasian populations, the light skin phenotype is the wild type now. Okay, it's normal for them now. If that makes sense, right? All right. That said, and keep in mind, this is pretty rare. You can have mutations that end up with a reversion back to the wild type, right? Keep in mind that all Caucasians are going to have only recessive genes for skin color. Some of them will have like one dominant gene for skin color. So if you took that population of Caucasians and you dropped them in the middle of Africa and came back 20,000 years from now, more likely than not, the skin would go back to being dark because the environment would favor the dominant gene for skin color. And well, the dominant genes for skin color. And those dominant genes would once again become concentrated within a small group in the population, which would have a benefit. And then over time, that small group would have more children in the larger groups. And of course, the skin get darker and darker and darker until the quote unquote white people turn right back to black, right? So that is a reverse mutation. And if you know about the peppered moth, which you should, you should also know that this is an example of reverse mutation. And we'll talk about the peppered moth later, okay? Now, you can also have mutations that are called missense or nonsense mutations, okay? See, this makes sense here. A missense mutation is caused when a base is substituted and it alters the codon, right? And it results in a different amino acid in the protein. So missense mutations alter the codons such that you get a different protein than the one you're expecting to get. So as it shows you here, TCA, this codon here, sorry, this triplet here, will give you, wait, this is not TCA the altering, wait, is this? All right, this is the triplet. This is the codon. Remember, it's codon in mRNA, well, RNA in general, and triplet in DNA, right? So AGT, when converted to mRNA, gives you UCA, right? And that codes for the amino acid serine. On the other side here now, you substitute AGT, and you end up now with AAT instead. So your mRNA is UUA, and this codes for the amino acid leucine, right? Significantly different amino acid. So if you get a change in the amino acids in the protein, it's caused by a missense mutation, right? Now, in the nonsense mutation now, you have changes in a codon into a nonsense codon. Nonsense mutations early in mRNA sequence produce a greatly shortened and usually non-functional protein. Now, you should know what a stop codon is, right? There's certain combinations of that let the ribosomes know when to stop what they're doing. Just to remind everybody, okay, y'all should remember this hopefully, right? That in order for you to make a protein, the DNA strand has to unravel, right? Then one of the two DNA strands is going to be used to make a mRNA copy of it, okay? The mRNA copy of that DNA strand is called mRNA, as in messenger RNA. That messenger RNA leaves the nucleus, goes into the cytoplasm. So far, this should all sound familiar to you, right? Then the mRNA settles within a ribosome, and then tRNA molecules, transfer RNA molecules, bring amino acids to the mRNA strand that are complementary and join them together to form a protein, okay? If that doesn't sound familiar, please let me know that I need to go through it in more detail, right? And so far, I'm working off of the idea that you know this stuff already, right? If not, please let me know. All right. Now, what this is saying is that making a change to the DNA causes a change in the mRNA, okay? On the mRNA strand, you're going to have a codon that lets the ribosomes, that lets the tRNA molecules know the protein is finished. Please stop adding anything more to it. Let the mRNA strand go. That is called the stop codon, right? In the nonsense mutation, the mutation that occurs does not cause a change in the amino acid that is being asked for. Instead, the mutation changes the codon from a regular codon to essentially a stop codon, right? Or something that convinces the tRNA to stop. So when you're reading the mRNA, trying to build your lovely protein, you hit the stop codon halfway through, and of course, you stop making a protein there, you kick out the mRNA. So now instead of having a functional protein, you have half a protein, right? That is a nonsense mutation. So obviously, if you're going to stop before you're finished, you're going to end up now with a shorter protein that usually serves no purpose. It is non-functional, okay? So it means, of course, that nonsense mutations can have severe effects on an organism because you're getting proteins that just don't work at all. Whereas missense mutations will affect the protein structure for sure, right? But only in one part of it. So the rest of the protein can potentially function. Go ahead, Daniel.
What happens to the protein since it can't be used?
Sometimes it builds up in the cell and actually damages the cells itself, right? Sometimes if the protein is important enough, let's say this occurs in a fetus that is developing, and the protein in question is an enzyme for the production, let's say, of ATP. Once you get a bad ATP production enzyme, all of a sudden that fetus doesn't have the energy to make the ATP it requires anymore, fetus dies. So you get a miscarriage, right? A lot of missense mutations simply result in the organism dying or not being born, right? They can be very bad. Though sometimes they can be good. We'll talk about that later, right? All right. So nonsense mutations, bad. Missense mutations can be bad. They can be indifferent as well, right? Or in some cases, they can even be good, right? But we'll talk more about that later. Then now we have silent mutations. Most of us have silent mutations, all right? We actually pick up mutations during the course of your life. So the average person, sorry, picks up about 3 new mutations every 10 years or so. So most of you are near the age of 20, which means you probably have about 5 or so mutations So you are now genetically different now than you were when you were born. Okay? But generally speaking, those mutations you pick up are referred to as silent mutations because the mutation itself does not result in any changes to you, right? So as it says here, silent mutations, they alter codons, but due to degeneracy of the codon, the same amino acid is specified. When they say degeneracy, what they mean is that you can have multiple codons coding for the same thing, right? And because of that, making minor changes to the codon or triplet, remember the triplet makes the codon, will not result in a problem, right? Y'all should remember the table. Where's the table? This table here should look familiar to you, right? And if we look, for example, at leucine, so we're looking right here. Leucine, you get leucine from CUU, CUC, CUA, CUG, right? So if you changed the last base here to any of the other ones here, you still get leucine. So that mutation doesn't matter. It's a silent mutation because you still end up getting the same thing. Same thing over here with valine, GUU, GUC, GUA, GUG. So any change to the end of this thing doesn't matter, right? As a matter of fact, usually speaking, changing the third codon tends to give you a silent mutation. That's the degeneracy they're talking about, right? So basically multiple different ways of getting the same outcome. So remember now, usually the last codon, if you change that one, if that one is mutated, it doesn't cause any problems at all. It's silent. Now the second one, the second codon now, second base, sorry. Notice here for leucine, for all of the leucine, the second base is a U, right? Which means if you substituted the second base, so instead of having CU, you have CAU, then what would you get? We have C here. Oh, sorry. That's what I want. C here for the first base, A for the second base. So come here. CAU, here we go. No man, the titles on this thing are wrong. This should be first. And that should be second. Yeah. Anyway. So CAU here gives you histidine, right? And CGU gives you arginine. So changing the second base causes a problem, even though changing the third base will not. Okay? All right, just to remind everybody again what I did is what I realized is that the labels we have here are wrong, okay? So this should not be second base. That should actually be the first base. And over here where it says first base, that should be the second base, okay? Just remind everybody how to read this table again in case you've all forgotten. If you're looking for a particular codon, let's say I want, I don't know, GUA. I want to see what amino acid GUA gives me. I come over here to the first base column to find my G. Then I have U as a second base, so I look up to find U. Here we go. So G and U converge right here. Then I can find GUA right there. And GUA gives me valine, okay? And I can do the same for any of the codons I can think of, right? Just remember, first base is on the left side, second base is on top for this one, right? All right. We'll get back to that table in a bit. Let's jump back to where we were before. So remember now, silent mutations normally occur in the codon at the level of the third base in the codon. Usually when you change the third codon, nothing happens. So you get the same exact amino acid. Changing the second codons is stickier. Sometimes it has an effect. Sometimes it actually doesn't, right? But it tends to lean towards having an effect. Changing the first base almost always causes a problem, right? All right. Then now we have neutral mutations. As this is a neutral mutation, is a mutation that alters the amino acid sequence in a protein, but does not change the function, right? Because the amino acid it gets is close enough to the one that was there before that it doesn't really have an impact, right? So for example, here, we have a change in our first codon. Well, not first codon, the first base in the triplet. Keep calling these codons. Remember this triplet up here, right? So instead of having GAA down here, which gives us the codon CUU, we have TAA. I remember TAA is going to change the amino acid. Changing the first one almost always does. So what we end up with now is AUU. CUU calls for leucine, right? AUU calls for isoleucine. Isoleucine and leucine are very chemically similar, right? So even though it's a different, technically speaking, that protein is going to be slightly different, it is still so close to what it should be, doesn't matter. Hence, the mutation is neutral. Yes, it's a change, but it doesn't matter, right? Then we have, of course, loss of function mutations. And when these happen, which can be substitution mutations or deletion or addition or any kind of mutation that causes your protein to have reduced function, is a loss of function mutation. So it causes the complete or partial loss of normal function of your protein, right? So your protein is so badly mangled or so badly truncated, it's chopped off, that it doesn't function the way it should, right? It mentions here that the mutation can occur in the regulatory region that affects transcription. So the mutation can occur while you're trying to make the protein using tRNA, right? So as you're translating, you're splicing the amino acids together, you're putting them together, they get put together wrong. That's your mutation there. And that, of course, causes the protein to be non-functional. It mentions here that is usually associated with a recessive condition, right? So loss of function mutations, remember I said to you, substitution reactions, frame shift mutations can all cause loss of function of a protein, right? But when we say loss of function mutations, we're specifically referring to this aspect here. When you have recessive genes resulting in translation errors in the protein, okay? So yes, everybody can result in a non-functional protein, but we usually use loss of function specifically for this, okay? I mention it because I don't want you to confuse them, right? Then now, rarely we have what is called a gain of function mutation. And gain of function mutations tend to produce proteins that result in new traits, right? And the traits are usually positive. So it causes a trait to appear in inappropriate tissue or inappropriate times in development. And these are normally dominant, right? Which is good because if you gain a function, right, that is good for your population, it means that you can more easily pass on that mutation to the rest of your population, right? All right, see if this makes sense to you. You know, we have hundreds of different breeds of dog, okay? You have some dogs like huskies. I'm assuming everybody knows what a husky is. If not, you can let me know and I'll put up a picture. All right. So we have huskies. We have sheep dogs. And we have like bulldogs. Now the reason I'm mentioning these three dogs is this. Huskies have a mutation that gives them a very thick coat with fine hairs. Okay? It is, of course, a dominant mutation. So huskies have naturally more insulation than most dogs. So huskies do very, very well in cold climates. So you can imagine dogs up in the Arctic being exposed to extremely cold weather. One of them develops a mutation for this very thick, fine fur. It's a gain of function mutation for them because they're in the right place for it. And because it's a dominant mutation, they pass it on to their offspring, and then all of a sudden, the huskies are born, right? Dogs adapted for very cold weather. Sheepdogs have long hair coats, but the coats are nowhere near as thick and fine because dogs come from England, the UK, which gets cold sometimes. It even snows there fairly frequently, but it's nowhere near as cold as the Arctic. So they have longer fur that is, and the fur is a little thicker. It's not as fine. So they don't do very, very well in Arctic conditions, but they can do quite well in the UK during UK's hot summers. During autumn, they can manage during winter. So having long fur being dominant in sheepdogs is good for them, right? Whereas bulldogs tend to have short hairs. Bulldogs are dogs that are designed for temperate climates. They don't do well in snow, right? So the hair length of these three animals and the fineness of the hair and the thickness of the hair is controlled by genes, of course. And depending on where the animals are when these mutations show up, they can be positive, okay? So see if this makes sense to you. If you had a dog in Jamaica that picked up husky mutations and the dog grew thick coat with very fine hairs, would that trait be a gain of function in Jamaica?
No,
Anybody?
sir.
Okay, Blessing said no. All right, here's the thing. It would still be a gain of function, right? But the function would be negative. It would be a net negative to the animal, okay? So the idea here is that the animal is now producing something new. Therefore, it has gained a function. Depending on where you find the animal, however, the function it gains could be a positive or a negative, okay? So gaining that function in Jamaica would actually be bad. But it would still be considered a gain of function because it didn't used to do it before. It does it now. You have gained something from it, therefore, right? And hopefully that makes sense to you, right? So you can have a gain of function mutation that is not necessarily a positive for the species based on when or where it occurs, right? All right. Then you have conditional mutations, which are expressed, as it says, only under certain conditions, all right? Which sounds kind of weird. Why would you have a mutation that only occurs under certain conditions? Let me give you an example that you are very familiar with, which of course is sickle cell anemia. The mutation in the sickle cell gene causes hemoglobin to become crystallized under specific conditions, which are conditions of low oxygen concentration. So the hemoglobin in some of the sickle cells perfectly normal until it gets into low oxygen areas, which considering these things drop off and pick up oxygen, is going to happen. But if you could keep that hemoglobin under nice high oxygen environments, they would not change. So this is a conditional mutation. It's a mutation that only shows up under specific circumstances, right? And as you know already, from lethal alleles, lethal mutations will result in the death of an organism, right? It doesn't have to kill them immediately, but right? But eventually it will end up shortening their life, right? Then now we have suppressor mutations. And a suppressor mutation is going to suppress the effects of another mutation. And it usually, as it says, occurs at a site different from the original site of the mutation. So organisms with suppressor mutation is a double mutant, but exhibits the phenotype of the unmutated wild type. Now, that sounds a bit weird, right? It's like saying, okay, the wild type fur of a brown bear is going to be brown, okay? Makes sense, it's called a brown bear for a reason. Some brown bear picks up a mutation that makes their fur gray, right? And then yeah. And then later on another mutation occurs within the gray bears that causes the brown color to return, right? All of a sudden you're saying, oh, that bear is brown. Technically it's a mutated gray bear. But you can't tell that because phenotype looks just like a regular brown bear, right? So that is a suppressor mutation. One mutation that stops another mutation from functioning the way it should, right? Now, so that is mutation based on phenotype, right? So now mutation based on mutagens, right? So remember, mutagens cause mutation, right? So causative agents of mutation. We have spontaneous changes. Basically, it means there's no specific thing that causes it. It just happens, right? The cells are replicating thousands of times. Sometimes you just make a mistake. It is what it is, right? Mistakes happen. Sometimes they're happy little mistakes. Sometimes they're not, right? So that's why I say spontaneous change to the DNA. Nothing necessarily had to intervene to cause the change. Which is, of course, where you will pick up most of the mutations you get during your lifetime. Remember what I said to you earlier? You're picking up around 3 mutations every 10 years or so. So your DNA is slightly different from what it was when you were born because of that, right? Then you have induced mutations. And these are, of course, caused by changes in the environment. So we're looking at chemicals, looking at radiation, right? So any environmental agent that can increase the rate of mutation, right, above the normal spontaneous rate is going to be a mutagen. So for example, high levels of UV radiation are mutagens, right? chemicals like benzene, for example, is a mutagen. X-ray radiation is a mutagen. Okay? It mentions here that the first discovery of a chemical mutagen was made by Charlotte Orbach, right? The basic idea is chemicals with structures similar to that of the four bases interfere with the bases, right? And polymerize with the chemical when you're forming DNA. So you're incorporating synthetic molecules into your DNA. So instead of having the AGCTU, you have some funny chemical that just happens to have properties similar to the DNA, to the base, but they're not going to function exactly like the base. So please keep that in mind, right? So chemical mutagens can be incorporated within the DNA itself, okay? So it gives here the example of 5-bromouracil, right? And 5-bromouracil is very similar to thymine in structure. But so if you look at the molecule itself here, you can see it is extremely similar to thymine. The major issue, of course, is this bromide group right here versus the methyl group right here. It's a methyl group, right? So the nitrogen is in the right place, oxygens are in the right place. Everything else is right, but this is wrong, right? So you'll end up incorporating this into your DNA, and then the DNA will not replicate properly. And so that's just bad, right? Now, any questions so far? All right. So now that we've gotten some of the background to the way, let's do a little bit more. Some of this stuff will be repetitious, but it's good to repeat to make sure we have the same, we have everything down. And that we think we understand what we understand, right? So let's go point mutations again. One second here. I need to change the background again. Okay. Right. So this is showing point mutations again. Remember, point mutations are substitution, insertion, or deletion within a gene. Okay. In substitution, remember what you're doing, you're swapping one nucleotide for another one. As we said before, you can have pyrimine to pyrimidine mutations, or you can have purine to pyrimidine, or pyrimidine to purine mutations as separate things, right? And the basic idea here is this. It shows you here a sentence. If you'll notice, the sentence here has words that's up in threes, because each word represents a codon, right? Well, so in this case, it would be a triplet. So the idea here is that when you take your original sentence, which is in the form of triplets, and you read the sentence, the fat cat ate the rat, the sentence makes sense. You get understanding from the sentence. So this is the equivalent of you making a protein from this this section of DNA, and the protein functions perfectly, right? You have full understanding. Now in a substitution reaction, of course, remember, we're changing one of the bases only. So if you read a sentence with a substitution now, We have the fat hat ate the rat. Obviously, the change is going to be right here. So instead of having a cat, we have a hat. Now, this sentence makes a lot less sense. You know, hats eating rats sounds weird, but at least you understand the words that are being said, right? So there's a change, of course. So this will make a protein that sort of works, you know, but so that it will not be completely useless. Now, if we take that same logic and we do an insertion mutation, remember, we started with the fat cat ate the rat again. In an insertion mutation, remember, you're adding now a codon that doesn't belong there. So if you notice here, the fat and the cat, for both of these sentences is perfectly fine. All is well, right? But this has been added, right? So now the sentence reads, the fat cat XLW ate the rat. What is XLW? Who knows, right? And in a deletion now, you're taking something away. So the fat cat ate the rat ends up giving you the fat ate the rat. So fat is just running around eating rats apparently, right? So just to hammer it home, and maybe maybe this is not necessary for me to do, but let's do one anyway. Let's do it from scratch, right? Just to make sure that we're all on the same page, right? So let us say we have a triplet like this. Let's say A, C, T, G, 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 A, C, A, C, A, T. Okay? So we have 4 triplets here. So this is our starting point. Okay? Now in a substitution one, we're going to take one of these letters and we're going to change it, and that's all. So A, C, T, G, 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 and let's say we change the A to a G again, so we get G, C, A, and C, A, T, okay? Now remember now, this is going to be used to make a protein. So which means you take this and you make mRNA from it. And when you make mRNA from this, you end up with, of course, UGA, CCC, CGU, and GUA. Okay? So remember, this is my triplet here. And this is my codons down here. Because this is DNA here and mRNA here, right? Now the mRNA is going to be used to make a protein. So let's quickly look back and see now what we can get out of that. So we have UGA. So let's find what UGA gives us. Remember, this is first over here, second over here. So ignore these, right? So UGA is a stop codon. Oops, that's a bad one. So we can have ACT here. Let's have AGT instead. That should be UCA, okay? So if UCA isn't stop. UCA serine, okay. So we have serine as one of the amino acids. And we have CCC, proline. I'm just going to write the abbreviations here. It will take less time. After all, you should remember what these things are, right? So we have serine to proline. And then we have CGU, which is arginine, right? Then we have GUA, and GUA gives us, wait, what am I doing? GUA, valine, okay? So this is what we get after the stop codon, right? So what does the original one give us? So remember, the original one is going to, so the AGT gives us UCA again, just like before, and CCC, just like before down here. And then we have GCA here and AC here. So ACA gives us UCU instead, right? So that is going to be the only difference we have. Just get rid of this out of the way real quick. So CCC, and this one here gives us UGU here instead of CGU. And then of course, CAT gives us GUA, right? So of course that means now in terms of the amino acids, we still get serine here, right? We still get proline there. But what does UGU give us? Hopefully it's not stop. Cysteine. So CYS. And of course, GUA gives us valine. Okay? So this is the original protein that we want. And this is the one that substitution has given us, right? So here's the thing. If we look at the properties of these two amino acids, okay? So we have cysteine versus arginine. Let's see what the properties are. So it says polar, non-polar, right? Aromatic, positively charged, negatively charged. So if we look up the arginine, so arginine here, arginine is going to be polar and positive, okay? I mean, if it's polar, so it's polar and positive. Now, if we look at cysteine, so cysteine, it is also polar and positive. This means that these two amino acids have similar characteristics, which also means that this mutation here might very well be a silent mutation. Because yes, the amino acid is different, but the properties are so similar that it might not make any difference at all to the final outcome, right? So far, so good. Y'all very quiet. Everybody okay?
Yes, sir.
Okay, good. All right, then now let's do a deletion one instead, okay? So remember, deletion is a frame shift mutation, okay? So remember, we're using the same thing here. That's where we're starting. So let's say we delete, I don't know, okay. So we have A, G, T, G, G, G. So let's delete one of these G's. We have A, C, A, and C, A, T, okay? Now, once I delete this G, we have a problem. Remember that our triplets have to be read in threes. So AGT, That's 3, right? But the next 3 becomes GGA. And the next one after that becomes CAC. And then at the end, I'm left with A and T by itself. Okay? So this codes for nothing at all, right? So AGT, of course, as usual, gives us the UGA for the code for the mRNA, and we get GGA here instead of GGG. So that gives us CCU. The next one gives us GUG, and then nothing there. Okay. So what do we get from this? UGA is a stop codon. Oh, sorry, it should be UCA, isn't it? It should be UCA. We had changed it to. Right, we had changed the UCA. So that should be serine, right? Sorry. That should be serine, my apologies. We had changed that one. So that should give us UCA, which gives us serine here. So that's serine. And CCU gives us proline. And GUG gives us valine. And we're 1 short, okay? So I want you to notice that all the amino acids after the point of deletion have changed. And we now end up with 1 amino acid short. So this protein here is completely messed up. Okay? So deletions can have severe impacts on the protein that you're trying to produce. But it happens after the point of the deletion. So you're shifting everything after deletion. That's why it's called a frame shift deletion, because once again, we're at the point of deletion, everything from there is shifted. Hence, frame shift, okay? If we do an addition for the same idea, so remember now we're starting with the same A, AGT, G, G, G. But let's say now I insert, I'm going to insert a new letter. I'm going to insert a T right here. Right? Then we have A, C, A, C, A, T. Okay? Now remember, once you reach here, everything else will change. So we still get back. So when we're doing this now for the triplets, we get AGT again. So we can say UCA again here. We get GGG again. So we can say CCC here. So far everything looks good. From this point on now, so instead of getting ACA, now we have TAC. Which means down here now we get AUG. And then TAC, we now have ACA. ACA. And that gives us, of course, UGU. And we have a T sticking out to the end there. And a U sticking out to the end there. Not doing anything with. Right? So now let us see. So we are at serine again, which is nice. We got proline back. We had lost it up there. Now, AUG gives us what? AUG is methionine. That's just, all right, so MET, fine. And UGU gives us cysteine, right? So once again, those last two amino acids are very different from the ones that should be there, okay? So whether it's an addition or a deletion, they're both frame shift. Everything after that point is altered. So the amino acids after that point are almost always going to be altered as well, okay? All right. Any questions so far, or this makes perfect sense? We can move on. Okay. I assume that means it makes sense. So that is gene mutation for you in a nutshell, okay? Now we can also have chromosome mutations, as we mentioned before. As we had said before, we used to lump chromosome mutations and gene mutations together and just call them mutations, right? Well, now of course we separate them. In a chromosome mutation, instead of having changes occurring to the individual bases within a section of the chromosome, which is known as a gene, you have whole chunks of the chromosomes with multiple genes that are being affected at the same time. Okay? So it's a more wide scale mutation that can affect multiple different phenotypes at once. Okay? So in terms of chromosome mutations, we have deletions, just like in the gene one. We have duplications, we have inversions, and we have what are called translocations. Okay? So deletions, duplications, inversions, and translocations. Now, does this make sense to everybody? Or should I use actual, or do I need to do this again? Does this make sense to you all?
Sir, can you do the other thing again, please?
All right, no problem. No, that's fine, that's fine. Okay, let's start with the deletion, okay? I'm gonna have to use a couple of different colors here. And I'm going to be using one single chromosome. Okay. So let us say I have a chromosome here. And let's say I divide my chromosome into sections. Right? Let's say 5 sections. So I call them 1, 2, 3, 4, 5. Okay, those are my pieces. In a deletion, here's what normally happens. Usually what's going to happen here is this. Your chromosome gets hit by some sort of radiation, or it could be a mutagen as well, and it causes the chromosome to break into chunks, right? So I have 1, 2, 3. Let's leave 4 and 5 together, okay? And by the way, this actually does happen. So when you're out in the sun and you're, I don't know, honestly I can't remember what most of you look like, but if you're light skinned enough, if you're out in the sun enough, you will get a sunburn, and that means the skin starts to peel, right? What is also happening to you is that the UV radiation from the sun is entering your skin cells. It's entering the DNA of your skin cells. It's slamming into the chromosomes and it's fracturing them into pieces, just like this. The good thing is that you have mechanisms to put these pieces back together, okay? Normally, of course, you get back your 1, 2, 3, 4, 5. But sometimes, instead, you get 1, 2, and then you get 4, 5 down here. Right? because 3 here got so damaged, it doesn't fit anymore, right? Because of the UV radiation mash it up so bad. So now you have a shortened chromosome. Remember, section 3 is not going to contain like one gene. Section 3 contains multiple genes, all of which now you've lost a copy of them, right? So obviously this can have some wide-ranging negative impacts on you because you're losing all of these genes one go, right? So deletions tend to be bad, all right? Then now we have duplication. And in duplication, we have the same scenario. Well, no, sorry, not the same scenario. In duplication, we have a DNA replication scenario. So you have 1, 2, 3, 4, 5 again, right? Then now you do DNA replication. Which of course means you're supposed to take this one and make a copy of it, right? 1, 2, 3, 4, okay. But sometimes you end up with the same section being copied more than once. So instead of having 1, 2, 3, 4, 5, you can have 1, 2, 2, 3, 4, 5. Okay. Now, hey, you're saying, oh, that's awesome. We have extra DNA. That's good, right? Here's the problem. When you get now, remember, this is duplication. We're doing known here. The problem now is that when you have this chromosome here, which has too many section number 2s, let's say this chromosome gets passed to a gamete, and then this gamete is the one that ends up in the zygote. Remember when you combine the egg and the sperm, the egg nucleus and the sperm nucleus have to combine as well, right? And in the chromosomes in the egg and the chromosomes in the sperm have to match up. Remember homologous chromosomes, right? So when this guy here now goes along, really should have changed the color of that one, but and it meets its counterpart from the female, let's say, okay? So here we have the female chromosome. And the female chromosome is going to have the same normal parts. So the female will have 1, 2, 3, 4, 5, just like the male one should, right? But this male one now, remember, has additional sections. So when it comes now and it tries to pair up with the female one, you're going to have a problem. Because now we have 1, 2, 2, 3, 4, 5. And you might say, well, what's the big deal? Well, they cannot stick together, right? But here's the thing. Section 1 genes are looking for section 1 genes to pair up with. Right? So they do that. Section 2 genes pair up with section 2 genes. Section 3 cannot pair up with section 2 because they don't have the same genes. Same thing with section 4 down here, wrong genes, and section 5, wrong genes. It means that these 2 chromosomes are now incompatible with one another. And when that fertilized egg tries to form the zygote, you will not get a proper alignment of the chromosomes and the zygote just dies, usually, right? So addition is bad because it doesn't allow for homologous chromosomes to pair up properly. All right. So far, so good, I hope. All right. Let's do the last two, and I guess we'll call it a day, which is inversion and translocation. So let's do another blank one. So remember now, we're going to be doing inversion and translocation. Now, y'all know what an inversion is, right? When you turn something upside down. Once again, this is going to be based on DNA damage. Okay? So once again, just like your skin cells, you get hit with UV radiation and your chromosome breaks into bits. So remember, we're doing inversion now. I mean, other things can do this, but we'll just stick to UV because it's easy. So here we have our chromosome again. Right? Then boom, here comes the UV. And we get our chunks. Let's say we get a chunk for 1 and a chunk for 2, 3, and another chunk for 4, 5, right? It wasn't quite as bad as the last one, right? Now, the mechanism for healing the chromosome comes along, and there are enzymes involved. We're not going to get too deep into that. And you're trying to put your chromosome back together. So you put part 1 where part 1 is supposed to be all is well. Then you grab the second section here, but you make a mistake, and you put it in upside down. So instead of getting 2, 3, you get 3, 2. And let's say you put the next section in, but you did a good job for that one, so you get 4, 5 down here. So now instead of reading 1, 2, 3, 4, 5, it now reads 1, 3, 2, 4, 5. Right? Sorry, not. Let's say this is not the skin. Let's say this happens in your gametes, right? Because that's where it's important. This happens in the gametes. You're sunning your balls or something. Who knows? Anyway, so we'll say mutagen here instead of UV, and we'll say benzene. Benzene, of course, is an industrial chemical that can cause this kind of damage, right? Now, we have, once again, the scenario of pregnancy. And once again, during pregnancy, remember, we have to align the chromosomes. So I'm assuming you can all see where I'm heading with this, right? So we have 1, 3, 2, 4, 5, right? So this chromosome from the male, because males are most likely to be exposed to benzene, tries to line up with the one from the female. They're the same height, so that's not the problem. But of course, section 1 loves to line up with section 1. That's great. Section 2 cannot line up with section 3 because it's the wrong section. 3 cannot line up with 4 either. But 4 can line up with 4. And 5 can line up with 5. Right? So now you have the same or a similar problem to you had before when the chromosomes, when they're trying to line up to form the homologous pairs, they can't attach properly because the sections don't line up because of the inversion. Okay? So that, of course, is bad. This can once again lead to the zygote simply dying, or it can stop the bivalent from forming properly. It can cause all kind of problems. Okay? So inversion, a section of the chromosome is flipped upside down. Not the whole chromosome, just a piece of it, which means that your genes are in the wrong place, right? Now, the last one we're looking at today is going to be translocation. In translocation, you actually need 2 chromosomes. Both from yourself, right? So we have ABC in this one. And in the next one, we have our normal 1, 2, 3, right? Now, you get hit with UV radiation again for this one, all right? And these two chromosomes, once again, break into chunks. That should be 5. Okay? Now, because they were busted up near each other, sometimes what will happen is that as you're trying to fit them back together, you mix up the pieces, right? So you get ABC over here. And then instead of getting the rest of the alphabet, you stick on a 3 down here, which of course doesn't belong. And then even if you put the rest of it back together properly, you know I have a chunk of the wrong chromosome sitting inside. And for the other one, of course, you're having the same issue. Right? So these two chunks do not belong. Okay? Now, this is actually very bad because once again, when comes time for you to replicate the chromosomes, so let's say you want to undergo mitosis. In mitosis, you have to have complementary base pairing and all that going on the same way, right? And then you have to have the homologous chromosomes pairing up. When chromosome number 1 here tries to pair up with its normal counterpart chromosome number 1, which has A, B, C, D, E, it can't pair up properly because that's A, B, 3, D, E. So that chromosome pair doesn't work out. And when chromosome number 2 tries to pair up with 1, 2, 3, 4, 5, you have a problem because you have 1, 2, C, 4, 5 instead. So instead of only one chromosome being affected, translocation affects 2 separate chromosome pairs, right? And the genes that you have now put in these chromosomes is very, very different from what should be there. They're literally coming from the wrong chromosomes, right? So the effects can be quite stark, okay? All right. Any questions? Or does this all make sense to you?
Yes, sir.
All right, wonderful. All right, it's 9:50 almost, so we'll stop here. So I'm hoping this makes sense, right? So next week we're gonna go through some types of mutation that we find occurring in nature, right? Oh, the rest of this is just types of mutation, so that's good. I think we have a class at 5 o'clock this afternoon, right? So therefore, I will see you all then, and have a good rest of your morning, folks. We will speak later.
Bye, sir.
Bye-bye.